Welcome to this discussion on transcription and translation. Here we are discussing how DNA is converted into RNA, which is the process of transcription, and how RNA is converted into protein, which is the process of translation. And we'll end with a discussion on how prokaryotic and eukaryotic transcription translational processes differ. So before we start, quick reminders on review of DNA and RNA. DNA is made of 4 nucleotides, A, T, G, and C. And if you take a human cell, there are 9 billion of them. These 9 billion nucleotides are arranged in random order in long linear stretches called chromosomes. There are 43 chromosomes. Each chromosome is millions of nucleotides long. For example, chromosome 1 is I think about 2. Well, let me not say that, but yeah, the chromosomes are very, very long sequences. Within those chromosomes you have stretches of A, T, G, and C that are that carry the code for proteins. In those sequences within the genome, that code for proteins are called as genes. So proteins are functional, responsible for the functional and structural characteristics of cells. And so enzymes, they make up enzymes, they make up muscle, they make up, they pretty much make us what we are. So the sequence of the amino acids that make the protein is critical. If there is an error in the sequence, then functionally we have deficit proteins, and the sequence of the amino acids are determined by the DNA, which leads us to the central dogma of biology. The central dogma of biology says that DNA becomes RNA through transcription, and RNA becomes protein through translation, and protein is responsible for traits, which are characteristics of an organism. So let's start with transcription. Quick review of DNA and RNA. DNA is a double helical, double-stranded structure. DNA molecules run, every strand of DNA runs from 5' to 3', and the opposite strand is anti-parallel to it. It runs from 3' to 5'. You have a sugar phosphate backbone within each strand, and you have nucleotides that are sticking in. And these nucleotides from the two strands are attached to each other by hydrogen bonds. A always binds to T using 2 hydrogen bonds, and G always binds to C with 3 hydrogen bonds. So A and T have, if you have a sequence of A's and T's, it's easier to kind of rip them apart because there are lesser hydrogen bonds than if you have a sequence of G's and C's, because they have more hydrogen bonds. RNA, on the other hand, is single-stranded for the most part, and unlike DNA, which has the deoxyribose sugar, RNA has the ribose sugar. The difference is that if you look at the pentose sugar of that makes up DNA and RNA has an OH group in the second carbon, and DNA has a H group in the second carbon. And RNA also has uracil instead of thymine, so it is AUGNC instead of ATGNC. Fun way of remembering this is all unicorns, AU, go to church. All unicorns go to church. And there are multiple types of RNAs. You have your messenger RNAs, your transfer RNAs, your ribosomal RNAs. All these three are involved in making in transcription and translation. And you also have siRNA and microRNA that are involved in post-transcriptional regulation and control. So transcription can happen from either one of those strands. So there are two strands running anti-parallel to each other. For a particular gene in the same chromosome, you could have a part of the upper strand acting as the template strand, meaning that that's the strand which is used to create the complementary sequence of messenger RNA, and a different gene could have the other strand acting as a template strand. So what we need to know is that strand that acts as the template, or the sequence on which your complementary messenger RNA strand is created is called as a template strand, otherwise called as the antisense strand, and the untranscribed DNA strand is called as the coding strand or the sense strand because it has the same sequence as the RNA. The enzyme RNA polymerase II is the key player in transcription. RNA polymerase II has multiple functions. It comes in, it opens up the DNA double-stranded structure to expose the single-stranded DNA strands, and then it helps formation of the complementary messenger RNA strand, and then it also winds it back. So it has multiple functions there. So the little bubble that is formed is called as a transcription bubble, and here you can see since this strand is what is used to make the messenger RNA, the strand is the template strand or the antisense strand, and the messenger RNA is synthesized in the 5' to 3' direction. We saw in DNA replication that DNA polymerase 3 also acts in the 5' to 3' direction. So that seems to be the same case here. So as we saw, if you look at a chromosome which is really, really long, millions of base pairs long, you have only small regions of this chromosome that are used for making proteins, and those are your genes, and not all of the genes are transcribed at any given time. There are about 20,000 to 30,000 genes in a human being, and there are about 100,000 plus proteins. So we'll talk about how that happens in a second, but we saw that not all 20,000 or 30,000 genes are expressed, meaning not all of them are made into messenger RNA and those are made into protein at any given time. Only some of them are. So how a gene is expressed or not expressed is determined by a sequence which is present before the gene. That's the promoter region, and the promoter region has as a series of T as and A's, continual sequence of T's and A's in a short stretch, which is called as the Tata box. So the Tata box is the place where transcription factors bind. Transcription factors are factors that are either activated by signal transduction pathways or if they are molecules like lipids, and they can pass directly from the outside of the cell through the plasma membrane and into the nuclear membrane and into the cell, and they can bind to this Tata box region. So the binding of the transcription factors initiates so the binding of the transcription factor into the Tata box in the promoter region initiates the process of transcription. The transcription factor binding causes a bunch of other proteins to bind to this as well, which finally recruit RNA polymerase II, which is the key player here. So this complex of transcription factors and other proteins along with RNA polymerase II is called as the transcription initiation complex. Once the transcription process starts, the transcription factors and all the other proteins leave. So only the RNA polymerase II is left behind, and RNA polymerase II elongates the elongates the RNA strand. So it carefully places the complementary nucleotides from RNA. So if it's a T, it places an A. If it's an A, it places a U. If it's a G, it places a C. If it's a C, it places a G. And then that strand continues to be synthesized. And as we saw, it is going to go in the 5' to 3' direction. The little bubble is transcription bubble is formed, RNA is synthesized, and then the RNA polymerase II closes up that bubble, and therefore leaving behind an intact double-stranded DNA strand the way it found it. Now, one of the things we need to see is there's a primary difference between eukaryotic and prokaryotic genomes. Eukaryotic genes have junk in them. When I say junk, it means or sequences that are not used, that are not finally made into protein. So if you look at the chromosome, for example, of a human, which is millions of bases long, the genes are very short. Sometimes they can be few thousand bases long, which means the rest of the sequence are not going to be coded. So those non-coded sequences are called as introns or intervening sequences. And these intervening sequences are also present within the gene, the start and end of a transcriptional unit. So and the sequences that are finally going to be used for making the protein, they are called as exons or the expressed DNA. So if you take a gene, for example, in this case, you have these blue sequences that are present, that are going to be made into the final protein sequence, and then you have the orange sequences here that are intervening sequences or introns. When transcription happens, from the beginning to the end, the whole sequence, including the introns and the exons, are going to be made. So that sequence that is created, the RNA that is created, that includes the complementary copies of both the intron and the exon sequence, is called as the pre-messenger RNA. The pre-messenger RNA is then spliced, meaning that all the sequences that are responsible for, that are not going to be used finally, are cut out. Okay, so and cut out, and then only the sequences that are finally needed are pasted back. So you can see here all the orange sequences, messenger RNA sequences corresponding to the introns, are cut out, leaving behind only the sequences that are coding for the final protein. So that final protein is called as a mature messenger RNA, which is a spliced RNA. And so the unit, the complex that does the splicing process is called as a spliceosome. Spliceosome is made of RNAs as well as proteins, primarily RNA. The RNA that does the splicing work is called as SNRPs, not Smurfs. And what they do is they form these lariat structures. And you kind of loop out the structure, which is the intron, and you cut it out and you excise it, and that excised portion is discarded, digested out. So that is one of the transcriptional processes that happens. Now, one other thing I want to point out here is that what is junk, or what is intron for one protein could be an exon for another protein. So for example, let's say this sequence here is an intron for this particular messenger RNA, which is going to be made into a protein. But the same sequence, the same pre-RNA, pre-mRNA sequence can be used used in a way that this portion is kept and it becomes a part of the final protein. So that way, the messenger RNA here, the final messenger RNA, the mature messenger RNA, is going to be different from this variation, because it's going to have that additional sequence of what was here, and that is going to be used to make into proteins. So what that means is we have the same pre-mRNA spliced out in different ways to create different variations of proteins or different types of proteins. So this is called as alternate splicing, which explains why we only have 20 to 30,000 genes but more than 100,000 proteins. So because of and one of the causative factors there is alternate splicing. Okay, so in addition to splicing, which is a part of RNA processing, there are two other major processes. One of them is called as capping. The other one is attaching a poly A tail. Capping involves adding a 7-methyl guanosine. So take a guanine and make add a methyl group to it and add it to the 5' end. That helps the cell recognize that this messenger RNA is not a viral sequence, so it won't digest it. There are enzymes called nucleases, endonucleases and exonucleases, that are floating around in the cell, trying to protect the cell from what it perceives as viral RNA and viral DNA. So when it sees a cap, a 5' cap, it tells, okay, this is part of our body, so let's not destroy it. So capping is an important part for protecting the newly synthesized messenger RNA. Another protective methodology the cell uses is to add this whole long sequence of A's, about 50 to 250 A's after the messenger RNA, and this is called as a poly A tail. The longer the tail, the more persistent the messenger RNA can be in the cytoplasm. So that's transcription. Now, all of the transcription process happened in the nucleus. The messenger RNA is now sent out into the cytoplasm using nuclear pores, which are small pores present in the nuclear membrane. And there, the messenger RNA meets ribosomes. Ribosomes are made of ribosomal RNA. There are the region within the genome which codes for ribosomal RNA is called as the nucleolus, rRNA, right? So rRNA is made by the nucleolar region. So the ribosomal RNA is a complex of proteins and RNA, and the messenger RNA that comes out seeks out and finds the messenger, seeks out and finds the ribosome. And in the cytoplasm, we have, let me quickly go through this. In the cytoplasm, we have the third type of RNA involved in transcription translation, which is the tRNA or the transfer RNA. So if you look at the tRNA, you can see that one end of it has the ability to attach to an amino acid. And being an amino acid, it's got all of the RNA sequences down there, right? So just know that the 3 nucleotides that are present here at the bottom, the other side, the 3 nucleotides that are RNA nucleotides that are found here are going to be critical in determining what kind of amino acid is going to bind here. So if this is a particular 3 nucleotide sequence here, then there is a corresponding amino acid attached to it. So these tRNAs along with amino acids are also floating around in the cytoplasm. So looking at our cytoplasm now, we have our messenger RNA that has come in, and it has attached to the ribosome, and in the cytoplasm we have transfer RNA or tRNA attached to amino acids. Okay, now biology has built a language for reading the messenger RNA, and what's beautiful is that whichever organism you look at, whether you're looking at a virus or a human being or a whale, it is the same strategy for reading the messenger RNA, which actually tells us that evolution happened, and all of these organisms, viruses and humans and everything, they were all having they all came from a common ancestor, simply because biology doesn't like to reinvent things over and over again. So here's the reading strategy. If you have a long stretch of A, U, G and C, the cell reads 3 nucleotides at a time. Okay, so these 3 nucleotides together are called as a codon. And so if you have 4 nucleotides, A, U, G and C, and you have 3 different, if you're looking at them in combinations of 3, you have a possible 4 power 3 combinations, right? 4 power 3. That is 64 possible combinations of triplets. Triplet meaning 3 nucleotide sequences. So you have 64 possible combination of nucleotides, 64 possible combination of codons that can be created. And each of the codons has a specific amino acid that it can associate with. Now when we say, or code for, when I say a codon codes for a particular amino acid, I'll go over in detail what that actually means. But that is where the whole magic of translation begins. So we have all these 64 different combinations of nucleotides, right? So there's 64 codons, 64 triplets that we have, and scientists have worked out which codon can associate or code for which amino acid, and they put this together in the form of a chart, and this chart is called as a codon chart. Now you can see there are some amino acids, some codons that are very special. So for example, you have AUG, which codes for methionine. So when you look at an RNA strand and you see an AUG, that's when the cell says this is the beginning of our translation process. Okay, so this is the start codon, and it codes for an amino acid called methionine. So PHE, SER, TYR, CYS, these are all names of the 20 different amino acids that are present. Now here's the thing, there are only 20 different amino acids but 64 different codons. What that means is there is going to be some redundancy, meaning more than one codon can code for the same amino acid. So you can look at this here. You can see that AAU and AAC are both coding for aspargine, ASN. Okay, and these four, ACU, ACC, ACA, ACG, they all code for the same amino acid threonine. And you can see that last one is the one that is kind of different, because that's easier to shift, right? So that third base change is called as a wobble. Anyway, so you have a redundant representation of amino acids for the 64 different codons. So we looked at AUG, which is the start codon. We also have 3 amino acids that do not have a specific amino acid that they code for. So they are called as stop codons, because when the messenger RNA has either of these sequences, UAA, UAG, or UGA, it basically tells that's a signal for the translational machinery to say we are at the end of our translational process, let's stop. And so we need to pay attention to that. All right, so let's come back to the transfer RNA. We said that there are 3 nucleotides at the bottom of the transfer RNA. Specific to that sequence, you have a particular amino acid attached to that. Now, if this transfer RNA with its random triplet anticodon triplet sequence comes by and finds the codon, if it's a match, if let's say if the messenger RNA has a sequence of AUG and the 3 nucleotides at the bottom here are UAC, then it's a perfect match, right? It's a complementary sequence. So but then you see that when it is UAC here, if the sequence is UAC, then the amino acid that is attached here is going to be methionine, MET. So the codon is attaching to the anticodon sequence, which is that triplet sequence at the bottom of the transfer RNA, and the transfer RNA is attached to methionine. So this specificity is what makes translation possible, that every transfer RNA has a specific anticodon sequence, and depending on that anticodon sequence, you have a specific amino acid that is attached to it. And because of the fundamental nature where you see a complementary sequence, you go and bind to it, this anticodon is going to go around. If it sees a messenger RNA and it sees that if it sees a match between its anticodon sequence and the codon sequence of the messenger RNA, then it's going to bind to it. And as it does that, it is going to bring its amino acid along. Okay, that's the whole magic of translation. Okay, so let's take a look at what happens to the messenger RNA and at the ribosome. So ribosome has 2 subunits. It's got a small subunit and a large subunit, and in the ribosome there are 3 locations that you can think of, right? Three regions. One is the A site, the other one is the P site, and the third one is the E site. A is for arrival or amino acyl site, attachment site. P is for peptide forming site, and then the E is for the exit site. So let's take a look at this, right? The A site is where let's start with the P site. Okay, the P site is where your transfer RNA with its amino acid is going to form peptide bond formation with the next amino acid that is coming in and going to sit in the A site. Okay, so think of this as a machinery, like a manufacturing table, right? So you make something and you keep moving this table. The table that is moving here is, or the thing that is moving here is your messenger RNA. So this messenger RNA strand is going to kind of slide between these 2 ribosomal subunits, and as it does, and happens, all of this happens really, really fast, you have a sequence that sits in the P site, you have another tRNA that comes and sits in the A site, and as it happens, there is going to be a bond that is formed between the amino acid that is sitting in the P site and the amino acid that is sitting in the tRNA that is sitting in the A site. And then the whole machinery gets moved forward by one triplet site. And what we need to know is that once we have a bond formed between the amino acids, then it releases from the transfer RNA. And then, so when the whole thing moves down, let me show this image. Okay, so here we are looking at the start of the translation process. Like you said, AUG, that's where it always starts. AUG is attached to methionine. When the messenger RNA has an AUG site, the anticodon site for that is UAC, and the UAC is attached to the UAC containing tRNA is attached to methionine, and so that starts that comes to the P site. That's the beginning, and then here you have the next three nucleotides as CUG. That's the codon. The anticodon for that is GAC, right? So the transfer RNA that contains GAC is always attached to leucine. We know this from the codon chart. So that's going to come into the A site. As soon as that happens, the methionine and the leucine, the two amino acids that are next to each other, they are going to form a bond. When they form a bond, again the whole thing is going to shift one more triplet. At this point, the methionine is going to lose its connection to the amino acid, the tRNA that was carrying it. So the tRNA is now sitting in the exit site or the E site. Remember APE, right? So it's going to sit in the exit site, and then at the exit site it leaves because there is no more amino acid attached to it. It has no value. In the same time, whatever is coming next to it, AAU, the anticodon, the tRNA containing the anticodon for it, with its associated amino acid, is going to come and sit here, and the whole thing is going to move one more time. So by this time, because so what happens is leucine is going to attach to this amino acid that is coming in, and it's going to lose its amino acid. It's ready to exit, so it moves over here, it goes. So this happens very, 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 very fast, in a way that as the amino acid keeps forming peptide bonds with each other, forming a peptide chain, the messenger RNA keeps kind of sliding through the RNA like a manufacturing roll, and then all the tRNAs that have lost their messenger their amino acid are going to go back into the cytoplasm, and a long strand of amino acid corresponding to the anticodon, which corresponds to the messenger RNA sequence, codons, are going to be created. So now you see how the DNA sequence transcribed into the form of an RNA, the same corresponding sequence, right? So that was there, and that sequence informed what kind of amino acid sequences are going to be created because of this language of codon and anticodon and the association between the tRNA and its corresponding amino acid. So overall, you can think of this as three different steps: initiation, elongation, and termination. So the sequence continues. This assembly continues until it comes to the either of those three termination codons, right? UAG, UGA, UAA. It's going to come to that sequence, and then it's going to say, well, I have nothing else to translate. So why that is because there is no transfer RNA that has an anticodon specific to any of these three sequences, UAA, UGA, or UAA. There are no corresponding anticodon sequences for those three codons. So therefore, there are no amino acids associated with that, and because of that, it comes to a standstill, and the translation machinery drops. That's termination. Now, the sequence of amino acid that is formed, which is the primary protein structure, is responsible for forming secondary structures. We saw that secondary structures of proteins are formation of beta helices and alpha pleated sheets. And then we have tertiary structures that form based again on the charges that are present in the side chains. Recall that an amino acid has 4 parts. It has got an H, it has got a COOH, it has got an NH3, and it's got a side chain. And the side chain can be polar or non-polar. It can be charged or not charged, positive, negative. Now all of these are going to determine how the amino acids are going to interact with each other, which means also that they are going to form different structures. So if I make a mistake, for example, if I have instead of having a sequence of hydrophobic amino acids and I replace them with a sequence of hydrophilic amino acids, I'm going to completely mess up the way that secondary and tertiary structures form and quaternary structures form, which means that the function of the protein is going to be modified. Which is why, again, we are going to see this later in the mutation lesson. We are going to see that there is, if there is an issue with the transcription, if there is a mistake in transcription, or if there is a mistake in translation, we are going to end up with proteins that are non-functional or not actively functional. And so the long primary chain, or the sequence of amino acids that is created using the messenger RNA as a template, is now transported across the endoplasmic reticulum, and it comes to the Golgi complex, where modifications are made by addition of lipids and proteins. And then if it is an excretory proteins, meaning that it has to be sent out of the cell, it is sent out through vesicles, or if it has to be kept back, used within the cell, then it is used within the cell. So let's run through the whole process quickly one more time. DNA is used as a template for making messenger RNA. The strand that is used to make that messenger RNA is called as a template strand, or the antisense strand. And the messenger RNA that is created is in eukaryotes has both pro and both introns and exon sequences inside, and those introns are spliced out using a process called splicing, and then whatever is remaining is the mature messenger RNA. The mature messenger RNA comes into the cytoplasm, where it meets the ribosome. In the ribosome, you have your transfer RNA and you have your amino acids. Transfer RNAs can bind to amino acids. This particular enzyme here, which is doing the job of binding the amino acid and the tRNA, is called as the amino acid transferase. And so what that does is the transfer RNAs have sequences at one end, 3 nucleotide sequences that are complementary to the 3 nucleotide sequences of the messenger RNA. So the 3 nucleotide messenger sequences of messenger RNA are called as codons, and the sequence at the end of the tRNA are called as anticodons. And the anticodon finds its codon partner and it binds. When it does that, it brings along the amino acid that it is attached to, and as the strand of messenger RNA passes through the ribosome, it kind of slides through in an assembly line, and as it does that, there is a formation of a peptide bond between the different amino acids that are brought in, and once they lose the amino acid, the transfer RNA exits the ribosome and goes back into the cytoplasm, where it goes and finds its corresponding amino acid, and through the amino acid transferase, amino acid synthetase, amino acid tRNA synthetase. Once again, the relationship between the tRNA and the amino acid is established, and then it finds its way back to make more protein. All of this happens very, very quickly. Now, very quickly, the difference between prokaryotes and eukaryotes in transcription and translation. One is that there are no introns in prokaryotes, and eukaryotes have introns and exons. The reason why we have so much more complexity in eukaryotic transcription and translation is because of the organism's complexity itself. Eukaryotes are very, very simple. Prokaryotes are very, very simple. They simply have they have very limited number of genes arranged back to back to each other in a circular chromosome, and they don't have any proteins associated with them. In eukaryotes, you have proteins called histones that are there, and they bind to the DNA and allowing the transcription machinery to either access them or not access them, which we are going to see in the next lesson on regulation of gene expression. So what is going to happen here is, in prokaryotes, since there is no nucleus or nuclear membrane, there's no organelles, right? As the messenger RNA is being made, the messenger RNA is immediately converted into let me see if I can. Yeah, there's a picture here. So as the messenger RNA is being made, the translation machinery also comes and starts making proteins using that messenger RNA. So you can see here is a beautiful picture. You have the DNA here, and you have your messenger RNA being made. You can actually see the messenger RNA strands, and then you can see the translation machinery is working just as it comes out of the factory, the messenger RNA factory, which is very different from what happens with the eukaryotes, because first it has to travel. You have to have all these modifications, RNA processing events like splicing and capping and poly-A tail addition, and then coming out of the cytoplasm, out into the cytoplasm and so on. None of that happens here. It happens very, very quickly. For that reason, because transcription and translation are happening parallelly, we have a much faster transcription translational process in prokaryotes compared to eukaryotes. So given all of that, we now have I think, a fairly good picture of how transcription and translation happens. We went through the process before. I would like for you to pause the video here and take a look at how label the different steps. What happens when and in what sequence, and which kind of enzymes are facilitating it, like RNA polymerase, and what is the APE site, and what the ribosome does, and what is capping, and all of that stuff. Just go through this whole process, run through it, and if possible, draw it out. That's the best way to learn. And hopefully this was helpful. Thank you very much.